Hello,
Hi.
Aaron.
Hey, how are you?
Great. So,
Thanks for coming.
thank you.
Do you want to give your slides a test?
Sure. So I just share screen, right?
Yes, please.
It works.
Looks good. Yep, looks good.
Okay. So I stop sharing or just leave here?
Stop sharing, please.
Okay.
I'm in Shanghai now.
Oh, that's good. So you get the air tickets.
Yeah, it's very expensive. I'm gonna be right back. I'm gonna just get a coffee, but I'll be right back in 2 seconds.
Sure.
Hey, it's on.
Aaron, you're back home now.
Yeah, back home.
Yeah. Sweet. Is Peng here yet?
I didn't see him.
Sure.
Wei,
And send him a message.
I sent him a reminder before. He said okay.
Okay. Hey, Wei.
Hey, nice to meet you here.
Yeah. Nice to meet you.
I know your name pretty often in MIT BBS.
Which was Oregon, right?
Yeah.
My team. Okay.
Yeah, that's something Aaron never know.
I know MIT BBS.
Oh, really?
Oh,
Yeah.
okay.
My wife was like a big shot on that board for
Oh.
pets, like a pet.
Oh, okay. Okay.
She was like a moderator of like a pet channel, a cat
Okay.
channel, even though she didn't have a cat.
Really? Okay. But I think dog might also counts.
No, there was a separate one for dogs.
Oh, okay.
That was like
the
precursor to WeChat.
Yeah, the modulator is very powerful over there. Can kick people out.
Oh yeah.
Yeah.
Hi, Aaron.
Okay.
Hi, everyone.
Hi,
Hi,
Paul.
Peng. Thanks for coming.
Thanks for hosting.
Yeah, of course. Can you test your slides, please?
Okay, let me see. Share screen.
There's a
Is
connection.
the slides, is the presentation all right?
Yeah.
Looks good. Well, yep, looks great.
Okay.
You're
Right.
the campus,
Outside.
yeah. I guess you got from PKU. The campus sometimes, but it starts okay. Yeah, but connection is a little bit slower than somewhere else.
Okay, I see.
That's fine.
Let me test maybe just the one video
Sure,
here
sure,
to see
sure.
it played
Yeah.
properly.
And sometimes you could use your phone internet if needed.
Okay, can you see the video alive?
Yeah.
Looks good.
Okay. I think previously I just flipped the slides too quickly, but when I speak, I will go through more slowly. So I think the connection is probably less an issue. But just in case, I have to turn on the hotspot on my iPhone, on my Huawei. Yeah. Okay, I will stop sharing.
Sure.
Yeah, looks good. So I was trying, Zilong, to I was trying to trick them so I could only have to stay in quarantine for 7 days, but it failed. I had to stay in 10 days.
At home for 3 more days, right?
No, that's not real. It's not seven. It's 7. It's 10 total. The rule is 7 plus 3, but no one does that. It's 10. Because
but still,
I heard, I heard in Pudong it's 10. So I told them I live in Shuhui, and then after 7, they said, "Where are you going?" And then I told them, and they said, "You can't leave." So I had to stay 3 more days.
Okay. Yeah, we should get together in the national holiday next week. Yeah, to celebrate, come back.
Yeah, I'm out. I'm gonna
and a barbecue beer again. Sweet.
Yeah, I'm ready. The food has been pretty bad in the quarantine, so I'm ready to go out and get
Yeah. You gained the weight you lost.
Yeah.
Yeah, so I guess the iOS meeting will be you can reschedule it on November sometimes.
Yeah, I think that's the plan. I'll try to figure out when. Yeah, it should. Yeah, I have to talk to. Yeah, I'll send a note to the group. Figure out that. We might want to wait until October to see what the situation.
Yeah.
We're also hoping to do a meeting in Hong Kong on non-human primates, but I don't know when that'll happen.
Oh, I haven't been there for a long time.
Yeah, same.
Just like 2016, right?
Yeah.
There's still be the UST. HKUST will be in the meeting.
Yeah.
Oh, okay.
Oh. Nancy Ip is the president now.
Okay. Yeah.
My dog came to pick me up at the hotel this morning. The quarantine.
Oh, okay. Oh, you just got out this morning?
Yeah, just this morning.
Oh, okay. Well.
Yeah. So they wouldn't tell me when I could leave, and then at 5 a.m. they just were banging on the door, telling me, "All right, you have to get out by 6."
That's funny time.
Yeah. Hey, Kong.
Hey, Aaron. Hey, Wei.
Hey.
Hey, good to see you.
Good to see you, Wei.
Hey, Pascal. Haven't seen you for years.
How are you?
It's okay.
Good. Look forward to your talk.
This is like a Sudhoff Lab month on NeuroZoom. last week was Manu, and then next week is Lulu Chen and Chenghui Pak, and then Jacqueline is coming.
It's just winning by sheer numbers.
I'm
just kidding.
And skill.
Don't tell Tom about this. I'm just watching Pascal like laughing away.
Yeah, no, it's not numbers. But if students want to eventually give a NeuroZoom talk, Sudhof is a good lab to go to get that.
Oh, let's get our.
That's shameless promotion, Aaron. Self-promotion.
Hey, Tom.
Oh, I'm here.
Tom is here. Sorry, we're talking not behind his back.
No, Tom, we were talking about how great your lab is.
Right, yeah. It's a pleasure to see you.
Yeah.
So, Wei, I haven't seen you for ages. I look forward to your talk. It will be good.
Thank you. I think last time it was a Brain Initiative Investigator meeting at the DC.
Well, that's ages ago.
Yeah, 4 or 6 years.
Yeah, a long time. Yeah.
Hey, Tom, how are you?
I'm fine, John. How are you?
Good. Long time no see.
Yes, very, very long time. Yeah.
Maybe one day soon we can all see each other in person again.
Well, why not?
Why not? I saw Lou at the Gordon conference. We should make plans to get together at some point.
Yeah, absolutely. Yeah,
wonderful.
We are full in-person meetings again. School,
yeah.
COVID, just
whatever.
Yeah, exactly. Who cares? Yeah.
Well, I care, but whatever.
China's not quite there yet, but getting there.
Yeah, they still have to reduce the quarantine by at least an order of magnitude or more, but hopefully soon.
Yeah. All right, Zong, maybe we'll start.
Sure. Yeah, go ahead.
Okay, hi everyone. I'm finally out of quarantine in a nice, comfortable Shanghai apartment. Good to see everyone for another NeuroZoom. We're continuing the Sudhof Lab Alumni Month, and we'll have a talk from Wei coming up. And then next week we have 2 more. We have Lulu Chen and we have Chenghui Pak. Both will give talks. Tune in. Let me or Zilong know if you want to present your research. You don't have to be from Sudhof Lab to give a talk, and we're looking for great science. So it's my pleasure to introduce our first speaker, Dr. Wei Shu. He's an assistant professor at UT Southwestern Medical Center. He did his undergraduate medical degree at Henan Medical University and a master's degree at Peking University. He then came to California, where he did his PhD at USC with Michel Baudry, and he studied glutamate receptors and cleavage products thereof. He then came to Stanford, where he did a postdoctoral fellowship with Dr. Tom Sudhof. And here he worked on tool development for studying circuits, but then he had a really interesting paper in Science, where he explored the phenomenon by which memories are stored, especially fear memories, and whether we store a general form of them or a specific form of them, and whether we do one or the other can have implications for how we can respond to situations that potentially dangerous situations, or whether we might get things like depression or PTSD. He described and identified a neural circuit that controls memory generalization and specificity. And really looking forward to hearing your latest Wei. Go for it.
Okay, thank you. Okay, first I want to thank the organizer for this great opportunity to see so many old friends. Today I want to talk about a work in the lab, which is we started on the coordination between the 2 processes of memory encoding and retrieval. So this is basically a dissertation work of a graduate student, Jun Guo, in the lab. He graduated last year, already went back to China. He's now in Shanghai, same as many of you guys, but unfortunately, he's no longer working in academia. So he did most of the work here for this study, and Suzy and Hinko was the 2 technicians in the lab, helped him with surgery and behavioral studies. Of course, we are neurobiology lab. We study biological memory system. But I want to start to discuss some human-made artificial memory systems. So this is the equipment invented around 150 years ago by Thomas Edison. So it's used to record sound. Of course, it's very simple. You just speak. The vibration of air will move a needle. The needle will move along the surface of a wax cylinder and make a grooves on the surface. So later on, if you want to reproduce this sound, you just rotate this wax cylinder. So it moves the needle and then regenerates the vibration of the air, so you hear the sound. So one thing I want to emphasize that the original prototype of this equipment was not really working very well, especially the sound disappeared after you reproduce a few times. And Edison spent about 8 months in the laboratory trying to figure out how to improve that. And eventually he figured out the trick. So it's extremely simple, is that when you record a sound, you use a sharp needle. So it moves along the surface, make the grooves. But when you try to reproduce a sound, you switch to another needle. That needle have a ball-shaped tip. So therefore, it moves along the groove without damaging it. So that's the trick. He made this machine work much better. So now, of course, this is a very old system, so we can move on to a new version. So everyone has this USB drive. So the memory elements of a USB drive is so-called floating gate transistor. Everyone of you have many of these, maybe billions of these in your pocket now. And it's also pretty simple elements. So basically, the way it works, you charge the floating gates with a voltage you put on the control gate. If you put a 10 volt voltage here, the electrons will be attracted to this floating gate, get it charged. When you try to read out the information, you put a 3 watts voltage on the control gate. If this floating gate is charged, that no current can go through this transistor. So this read as 0. If the floating gate is not charged, you can have a current that goes through this transistor. It's a logical one. So therefore, you can remember, keep a lot of information in this system. Of course, it's a modern memory system. From these two human-made memory system, we can see some common features of the memory equipment. First, to make a memory, you have to make a physical change in the system. Either the grooves on the surface of a wax cylinder or the charge of a floating gate. You have to make some changes. So for the biological system, we still don't know what's the physical change is really related to memory, but the synaptic plasticity is clearly the most important candidate for that. Another important thing is that the encoding, if we use this word for write information into the system, and the retrieval is reproduce the information. These two processes have to be converged on the memory traces, basically the physical change of the system. So that's basically no other way you can do that. So in recent years, there's a lot of study on so-called engram, basically kind of support this idea. It's basically the same neuron which get activated during your appearance of something that get reactive when you try to reactivate that memory. So another thing, the third one, which is something I'm going to talk today, is that encoding and retrieval, although these two processes need to converge, they have to be physically different when they reach the memory trace. Otherwise, as we just showed by the phonograph, you either damage the memory or you cannot retrieve that. So then the question is, how in the biological system, how we get these two processes physically differently and separated. So now we come to the hippocampus, our favorite brain region studying linear memory. So as you all know, the hippocampus is very laminated structure. The pyramidal cell of C1 that attend the apical dendrocytes towards dendritic gyrus. It received two major streams of excitatory synaptic inputs. One mainly is distributed in stratum radiatum, we call SR, that's easier to remember. That synapses mainly come from C3 region of the hippocampus. Another layer we call SLM, which mainly receives the input from entorhinal cortex to a less degree from the midline thalamus, especially nuclear reunions. A few years ago, actually 20 years ago, that's a nice model was proposed how the memory encoding and retrieval can be coordinated in the hippocampus C1 region. So the idea is that at a certain phase of Z oscillation, at that moment, when the entorhinal input to C1 is strong, but the C3 input to C1 is weaker, at that moment, the C3 to C1 synapse have high potential to have plastic changes. In that time, you encode memory. But at opposite phase of the zeta, in that moment, the entorhinal input to C1 is weak, but the C3 input to C1 is strong. At that time, C3 to C1, the plastic change is unlikely to happen. At this moment, you retrieve memory. So this is a nice model, but I get my training mainly in synaptic physiology and biology. So I have difficult to really understand the point here, because why plasticity between C3 and C1 preferentially happen in a moment the synaptic input is weaker. Because we know the LTP principle is that there's a cooperativity between synapses. When there's a lot of synapses acting at the same moment, you have a better chance to induce LTP. But why C3 to C1 synapse can have higher possibility to have plastic change when the input actually is weaker? So that's the question we want to address today. So now we come back to this C1 circuits. So many years ago, because we are studying the input from the thalamus to the C1, so that's a hypothesis that the major impact from the thalamic input on hippocampus mainly through interneurons. So therefore, we searched the cell types, the interneural cell types in this particular layer, the SLM layer. So we did a RNA scope and checked a few markers of interneuron. We found that the ending NF is a marker preferentially expressed in SLM layer and account about 50% of the cells in this layer. And other major interneural markers like NPY, CCK, or NOS1, they have more broad distribution in the hippocampus. Therefore, we try to target those NDF positive cells in the hippocampus. So we get a Cree driveline from Allen Institute. I believe that's already the third version of this Cree driveline. But still, when we cross the reporter or we inject a virus to the Cree positive mice, we still see a lot of expression in other neurons instead of the interneuron in SLM layer. And fortunately, around that time, there's a paper published describe interneuron specific promoter. So we put those interneuron specific promoter to drive reporters in combination with this Cree driveline. And with that, we can achieve pretty specific targeting of those NDF positive interneuron in SLM layer of the C1. So now we can target those cells. Of course, we can do some basic characterization. So those cells, majority of them have pretty dense but short processes. That's a kind of morphological feature of a neuroglial cell. And if we patch those cells and inject current to try to fire them, and the majority of them have so-called delayed firing property. Basically, when the injected current is kind of close to the threshold of firing, it takes some time to let the cell really fire. Another feature is so-called spontaneous firing. Is basically you inject current repeatedly for a few times, then you just stop inject current. Those neurons can continue to fire for tens of seconds or even minutes. So majority of the neurons labeled have those properties, and all these properties fit the features of so-called neurogliaform cells, suggests that those NDF cells are primarily neurogliaform cells of CE1. So then we did some analysis with their connectivity using the G-deleted rabbit virus. So as you can see, as I packed it, we get a lot labeling in the entorhinal cortex, suggests that entorhinal cortex is indeed a major input to those neurons at SRM. But beyond that, we also get a lot of cells labeled at CE1 and subiculum, and even at CE3, suggests that they also get some feedback input from either CE1 or subiculum. And they also seem modulated by the acetylcholinergic input from the septum. So to know their function, so the first thing we did is put a GCAM in those cells and to check how when they fire. And because those cells actually pretty sparse when we implant the granulins to monitor their activity, in each animal, we normally can only get 3 to 5 cells. So if we put all the calcium traces together, you can pretty easily see a pattern is that first, they have some basal activity when the animal was in the home cage. But when you subject those animals to a field conditioning training, you can see overall speaking, the active calcium events become more frequent. And if you put the animal in the context test, basically you put it back the animal to the original cage, they get shocked. So now you can see the overall calcium events become much sparser. And in the third day, if we put the animal to a different context, so the animal start plot again, now you can see the calcium events become increased again. So this data suggests that the activity of those neurogliaform cells are positively related to training and negatively related to retrieval processes. Of course, this is just correlation, and we want to check if there's a causal relationship here. So what we did is that we put excited dread into those cells and to check if we manipulate the activity of those cells, if they change the memory. So first experiment we did is that we inject clozapine to activate those cells before we training the animals in field conditioning. So then when we tested the animal, we found that the Cre-positive cell, the basic Cre-positive animal, basically activate those cells, they remember better than those Cre-negative animals. Suggests that if we stimulate those neurogliaform cells when the animal is learning, we help the animal to remember. To as a control to make sure this specific effect from those neurogliaform cells, we also run a control experiment. In this control experiment, we basically impress dread in all interneurons in the CE1 area. If we activate those cells during learning, what happens is that memory kind of get impaired. That's consistent with most people believe that hippocampus is indeed important for contextual field memory. And it also proves that when you activate those neurogliaform cells, you can achieve specific effect that is facilitates memory encoding. So then we use another common used behavior test to check if this consistent across different behavior paradigms. So this is a novel place preference test. As you can see, if we inject clozapine before training, we basically get the same thing, the animal learn better. So now we want to switch, change the way we run this experiment. Now we no longer inject clozapine before training. Instead, we inject clozapine right before recall. And what we found is that if we activate those neurogliaform cell, you get the field conditioning impaired. Same thing for place recognition test. If you activate those neurogliaform cells, the memory was impaired. So now if you activate those cells during learning, you help memory to encode it. And if you activate those cells during retrieval, you impair the memory. So what happened? So now we want to look at the C1 pyramidal cell, because we don't think those interneuron carry those memory. The pyramidal cells are more likely the real players for encoding those memory. So what's the impact on those pyramidal cells when we activate those neurogliaform cell? So what we did is that we put the excitatory dread in those neurogliaform cell and monitor the calcium activity in the C1 pyramidal layer. So as you can see now, you can see record much more neurons. So first is that we just did some recording in the home cage to see if we activate those cells can produce any impact. As you can see, if we compare a Cre-negative and Cre-positive cell, after injection clozapine, within 10 minutes, you can see a significant reduction in the neuronal activity, suggests that with DREAD, we can indeed activate those interneurons, which reduce the activity of pyramidal cells. So then we subjected those animals to field conditional training. And you can see the first 2 minutes, that's basically the time animals spend the time to explore the cage. You can kind of think that's the time animal really learn the environment, is encoded memory. So at this moment, you can see overall speaking, the Cre-positive animal have lower activity. That's consistent with home cage data. So you activate inhibitory interneuron, you reduce the pyramidal cell activity. So what happened is after the shock is different. So in the Cre-positive cell, Cre-positive animal, you can see that's significantly higher activity in the pyramidal cell compared with the Cre-negative animal. So basically the relationship is inverted now. So you activate a group of interneuron in the hippocampus, you actually facilitate the firing of C1 pyramidal cell. So that's kind of unimpacted. So now we want further go to the local circuits to figure out what's happening. So we basically put a channelodopsin in those NDF positive neurogliaform cell and shine light to do electric field recording to see what happened. First, we did a field recording that's pretty impacted, that we only see inhibitory potentials in the SRM layer because most of the fibers is in this layer. And then we did a local field recording with stimulation applied to different input to the pyramidal cell. So first, if we stimulate SRM layer, the interneuronal inputs, what we found is that if we activate the neurogliaform cell with light, it reduces the excitation from interneuronal cortex. That's also impacted because you put an inhibition in this layer. What's unimpacted is that if you stimulate the C3 input at SR layer, at the same time you stimulate those neurogliaform cell, what you see is that the postsynaptic potential actually was enhanced. Because the actual cellular recording is not that, it's kind of noisy. So we switch it to whole cell recording here. So we patch the pyramidal cell and shine light on the neurogliaform cell. You can see the red trace, that's inhibition phase after you use light to stimulate those neurogliaform cell. And then you have electric stimulate EPSC. And if that's excitation of neurogliaform cell, you can see the red trace, actually the excitation phase was enhanced. That's consistent with the field recording, suggests that if you activate those inhibitory neurogliaform cell, you actually facilitate CE3 to CE1 synapses. So how can that happen? So one possibility is that that's a disinhibition happening. It's basically those neurogliaform cells inhibit some interneuron at the SR layer. As a consequence, you disinhibit the CE3 to CE1 input. To test if that's the possibility, so we patched those interneuron in SR layer. So we basically randomly patched the interneuron in SR layer. Actually, in 100% of the cell we patched, they all get inhibitory currents recorded from when you excite those neurogliaform cell. suggests that indeed those cells can disinhibit C3 to C1 input. So now it's kind of clear. So those neurogliaform cells inhibit one pathway, but disinhibit another pathway. So this suggests that if at a moment, pyramidal cell is primarily activated by entorhinal input, so those neurogliaform cell activation will reduce their activity. And if the pyramidal cell are primarily driven by C3 input, you can imagine if you activate the neurogliaform cell, you will make a pyramidal cell more easy to fire, right? So if we match with this data, suggests that during the first 2 minutes exploration period, the pyramidal cell are primarily driven by entorhinal inputs. And after the shock, the pyramidal cell are primarily driven by C3 inputs. And if that's true, so we did imaging. So we basically impress GCAM at either entorhinal cortex or contralateral C3. So we have GCAM impressed in the different layer at C1, and then we implant green lens to monitor their activity respectively. And you can see that's calcium fluctuation associated with animal behavior. But that's a caveat here is that the entorhinal calcium signal is much stronger than the C3 calcium signal. So we have to do some normalization to do the comparison. So therefore, we normalize the traces to the first 2 minutes. After that, you can see after the foot shock, that's a period the C3 clearly have an increase over compared to entorhinal cortex. This consistent with the data we obtained with the pyramidal cell activity. So now if we put everything together, we can come up with this model. So basically one animal is explorer environment, try to encode the memory, you have entorhinal input, have a strong action in C1. At the same time, the active neurogliaform cell. As a consequence, this neurogliaform cell will disinhibit the interneurons at SR layer. As a consequence, disinhibition is a very good way to allow plasticity to happen. Therefore, you can produce plastic change. Therefore, you keep memory in this system. So when memory is going to be retrieved, the entorhinal input is weak, and the C3 input is strong. As a consequence, the neurogliaform cell is quiet, and the interneurons at SR layer is strong. Therefore, those synapses are and strong inhibition. That's not much plasticity to happen, but because of strong C3 inputs, they can fire those cells to retrieve memory, but without changing memory. Basically, that's the model I want to present today. I guess I can stop here, and we'll be happy to answer any questions.
Great, thanks, Wei. Awesome talk. And now we're open for questions.
It looks like I did not present the controversial stuff.
Maybe everything is super clear and compelling.
Hey, wait, I got a quick question about the input of the neurogliaform cells. So you're suggesting from multiple places like septum. So you in order to encode the memory, don't you think they have to come from some specific sources to regulate? Yeah.
So our tracing data indicated that the received inputs mainly from entorhinal, but also from other regions.
Okay.
But from this, electrophysiology analysis, we show actually entorhinal is clearly the strongest.
Okay. Oh, okay. So I was thinking, why they need to bypass to reinforce the C3, C1 circuitry, right?
They did not bypass. They just switch the two pathways.
So
during encoding, they make one pathway strong. During retrieval, they make another pathway stronger.
Okay. Yeah.
Okay.
Yeah. So somebody returned.
All right, Meow.
Hi, Wei.
Hey,
Great talk.
Neuro.
Great talk. So I wonder, because you mentioned ganglion cell at the beginning, have you tried to measure the connection between these cells with ganglion cell?
We did not. You mean let's try to figure out. So we will think. So okay, another feature of those neuroglial cell is that they have so-called volume inhibition. It's basically they don't have a typical synapse. The GABAergic synapse is kind of non-typical. It's basically release GABA to diffuse to quite a large area to produce a kind of global inhibition. Therefore, I don't think they have such kind of special precision to just inhibit one type of, engram cell or not enabling cell. It's more like just switch the two pathways and make a pyramidal cell at a certain moment easy to either encode or easy to retrieve information.
Okay, thank you.
Kai-Wen?
Hey, Wei, great talk.
Hey.
So I have a few questions regarding this very special SLM neuroglial cells. So it seems like this type of inhibition neurons are kind of not that much abundant, right? So it's not like a lot of this SLM inhibitor neurons in the hippocampus. How does that how their inhibitory outputs compare, how the strengths or the potencies of this type of inhibitor neurons compared to the other cell types in the inhibitory cell types in the hippocampus? Seems like it's like it can actually it can have a very potent inhibitions to control the CA1 neurons activities at a very time accurate manner. Is that what do I understand your talk correctly?
You mean compare with other interneuron in the hippocampus? Is it stronger or weaker?
Yes.
Is that I don't know how to compare that because they have a very specialized, localized innovation in the hippocampus.
But
so
when we, I think my question.
When we patch it, okay, go ahead.
No, you finish first, please.
Yeah, so okay. So when we patch the whole cell recording of pyramidal cell and shine light to stimulate those neuroglial cell, we can see clear iPSC. And that's iPSC from the very distal part of the dendrites. I think that suggests to us that inhibition is pretty strong already. Because if we record the excited synaptic current from interneuronal input, actually pretty weak.
Okay, so that's basically that kind of answer my questions. So I think my question is, if you record the evoke iPSC in C1, and when you blocked this neuroglial cells, how does that will affect how much it will affect the magnitude of the iPSC? That may kind of estimate the contribution of this type of inhibitory neuron to the inhibitions to CA1 neurons.
Okay, I see. So your suggestion is that you use electric stimulation applied.
I'm just curious. So because we are recording iPSC all the time, so we don't know where this iPSC comes from, right? So maybe the SLM neuroglial cells may contribute a great proportion of the inhibition to the C1 neurons.
Okay, yeah, that could be an interesting experiment to do, but we never did that. You mean just electric stimulation, but use optical silencing to see the impact?
I'm just curious. And another question is regarding the activation pattern of this type of inhibition neurons. Seems like it's only determined by the inputs. So when they receive the inputs from the entorhinal cortex, they activate and control the encoding. When they're not receiving the inputs from EC, then they will remain silent when the animal need to retrieve the memory. Is that correct? Or
that's what we think, because entorhinal is the major input to those cells. So it looks their activity. I mean, from all our recording so far, their activity is kind of correlated with entorhinal activity.
Okay.
Yes.
And there are also many other factors that can regulate memory encoding and retrieval, such as like the brain states, like neuromodulations, different type of
because like acetylcholine, it says how they interact. We don't know, because now it's still pretty early stage. So, but I will say interneurons that tended to work at a much faster time scale compared to neuromodulators. So that's if the model I presented by Hasmore, if that's correct, you need to switch the encoding and retrieval in a very fast time scale. So that interneuron could do that work maybe better than modulators.
That's true. So I mean, the neuromodulator may controls the excitation state of this type of SLM interneurons that predetermined how the animals can encode and retrieve the animals.
Yeah.
Is it? Yeah. Okay. Thank you. It's great talk. Thank you.
All right. Awesome. Wei, thank you very much.
Okay, thank you. So I guess I can.
Zilong, you can introduce Peng, please.
Okay, my great pleasure to introduce Panzo from Peking University. So Peng was trained as a chemist, so graduated from a chemistry major in Peking University, a bachelor degree. So after the bachelor degree, he went to MIT to work with Alice Tin, also a kind of interdisciplinary chemist to make a cross to the biology. So in PhD in MIT, he started interested in making all kinds of chemical tools to facilitate biological researches. So after PhD, Peng went to Harvard, worked with Adam Cohen, where he started to use the chemical tool to develop all kinds of neuroscience tools, including the voltage sensor and a lot of imaging tools. And to 2015, so Peng come back to Peking University to start his own lab. So in his lab page, he said illuminate neuroscience, which is also his dream, and also I believe will make a great impact to neuroscience like us. So today Peng will talk about his latest in his own lab. So hybrid voltage indicator for mapping neuronal action potentials. Welcome, Peng. Take it away.
Thanks, Zilong, for the very kind introduction. And I also want to thank Aaron for the kind invitation to giving me the opportunity to present my work. Let me start the sharing. And today I want to tell you about our recent progress on developing genetically encoded voltage indicators. We all know that electro-based recording, such as patch clamp, has been the gold standard for measuring neuronal activity, such as recording action potentials. But a common problem associated with electro-based recording is that they lack spatial resolution. Typically, you just record from one neuron at one spatial site using one electrode. This is relatively invasive, and as a result, you have very low throughput and low spatial resolution. But the electro-based recording has very high speed and sensitivity that is unrivaled by any other methods. One idea is to get rid of the electrode and try to convert the electrical signal into an optical readout. And if you want to measure electrical activities from a neuronal population or try to record electrical activities from multiple sites to understand how the spatial arrangement and the dynamics of actual potentials, then you need to put increasing number of patch pipette onto your electrophysiology rig, which has been a very daunting task as been illustrated in this figure. Then how to visualize the membrane voltage? By definition, this requires developing an electrochromic sensor, which can sensitively report electrical signal using an optical readout. The sensor must be sensitive enough to resolve millivolt level voltage changes and have sufficient temporal resolution to report actual potentials at the millisecond time scale. We chose a rhodopsin protein from the algae called Acetabularia acetabulum, and my graduate student has been working on this for the past few years. Originally, this rhodopsin functions as a proton pump, meaning when it's illuminated with a light, it can pump one proton from inside of the cell to the outside. For this type of proton pumping rhodopsins, previous work in Adams-Cohen's group at Harvard has established an electrochromic property for these proteins, meaning the voltage fluctuations can drive the reversible protonation. This is because at the center of the protein, there's a retinal sheath base that can be reversibly protonated. When the protein is sitting on the membrane, the electric field will pull on the positive charge proton, causing either a facilitation or inhibition of the protonation, which then results in a change in the absorption spectrum. This can be even quantitatively described by the Nernst equation, as the physical chemist would be familiar with, which means that this 60 millivolt voltage change will result in a perturbation in the proton affinity, such as the pKa, by 1 pH unit. Then the next challenge is, even if we understand this voltage fluctuation can drive a change in absorbance, how can we measure the absorbance very sensitively under the microscope? One idea is to convert the absorbance into a very sensitive fluorescence readout, such as by putting a fluorophore in close proximity to the membrane-embedded rhodopsin. And this is what we call electrochromic FRET. And back in Adam Cohen's lab, I've been working on this problem. And back then, we were using a fluorescent protein fusion to the rhodopsin, which to demonstrate that the electrochromic FRET could really work. And more recently, at the Genelia farm, the Voltron has been engineered based on exactly the same principle. And we want to improve the sensitivity of electrochromic sensors. So we want to get rid of the fluorescent proteins or the halo tag protein tags. And we want to directly conjugate a small molecule fluorophore to the protein on the membrane so that the fluorophore and the retinal sheath base will have a very short distance between them. And we can maximize the FRET efficiency in this way, and which then could lead to increased voltage sensitivity. So the basic idea is illustrated in this slide. And the fluorescence can be stimulated by the laser excitation. Because there's a FRET energy transfer or FRET quenching mechanism between the fluorophore donor and the retinal in the rhodopsin, the dye emission can be modulated by the amount of rhodopsin absorption. When there's a membrane depolarization, the proton is attracted to the retinal sheath base, causing an increase in the absorption and a concomitant decrease in the dye emission, which leads to the formation of the electrochromic FRET. We can simply monitor the very bright fluorophore as a readout of the absorbance, which in turn can tell us information about the membrane electric field. The challenge is how can we conjugate a chemical, small molecule, very site-specifically onto the rhodopsin on the cell membrane. And this has to be done on a live neuronal surface. And we turn to chemical ligation tools developed by a chemical biologist lab. And there is a ligation, there's a ligase that can catalyze the conjugation between a vitamin called lipoic acid onto a short peptide. This enzymatic ligation will use the small molecule and take the ATP to form an adenylate intermediate, which then is conjugated to the lysine side chain of a short amino acid, a short peptide. by mutating the active site gatekeeping residue in the bacterial ligase, in the past, other labs have demonstrated that this could greatly expand the binding pocket of the ligase so that could tolerate a new substrate. And we chose a very bulky new substrate containing an 8-member ring called trans-cyclooctene. We chose this molecule because the trans-cyclooctene is a highly strained molecule, and it contains a trans carbon-carbon double bond in a very small 8-member ring. Whenever there's a chance to release this strain, it will react very rapidly. So we take our rhodopsin on the membrane, we will insert a 13 amino acid, a short peptide loop into one of its extracellular loops. Then in the very first step reaction, we use the engineered bacterial ligase to conjugate the trans-cyclooctene moiety onto the protein surface site-specifically. And then we will add in a tetrazine conjugated small molecule. Between the tetrazine and the trans-cyclooctene, they can react in a way called the Diels-Alder reaction. So we can conjugate a small molecule very site-specifically onto the protein using this very simple 2-step ligation process. Once this is done, then the membrane voltage will shift the rhodopsin absorption, which will then modulate the Fred efficiency. In this way, we could conjugate a hybrid voltage indicator. It will use a small molecule fluorophore reporter and a genetically encoded protein backbone to achieve a cell type specific targeting. And this would work on the live neurons because the Diels-Alder reaction between the tetrazine and trans-cyclooctene is very fast and highly biocompatible. Under the microscope, we could indeed see that when we stimulate the neuron to fire using the electrode, we could record the firing of the action potential from every single pixels under the microscope. And we use this HVI, the hybrid voltage indicator, to achieve continual recording of neural activity over the time course of tens of minutes without really losing much signal-noise ratio. This is because the typical organic dyes are much more brighter and more photostable than fluorescent proteins used in most labs. And we could see an increase in the excitability when we throw in the gamazine to stimulate excitability in the cell culture. Or we could try to block synaptic transmission by adding in different blocker APV or MPQX to see the decrease in excitability. We could also try to, using different chemicals to modulate the firing of action potential. For example, when we treat the cells with cyclothiazide, we could see that excitability will slightly increase, and then the firing pattern will shift from the previous tonic pattern into the burst-like activity. This is consistent with the previous report. So all of this to demonstrate that we could indeed use HVI to monitor neuronal action potentials with a very high spatial resolution, and we can do this stably for a very long period of time over the course of tens of minutes. And then we could choose different fluorophores. We could expand the palette of our voltage indicator by simply replacing the fluorophore from red, orange, red to the far red. When we use the far red indicator using the Psi5 as the reporter, then we have created space for the spectral space for other purposes. For example, we can use the blue light to stimulate the neuron optogenetically and use yellow light to inhibit it optogenetically using two light-sensitive ion channels and pumps called the Sheriff and the MAC. In this way, we could do the reversible excitation and bidirectional stimulation, while at the same time, we could monitor the neural activity using under the microscope. In this way, we could completely get rid of the requirement of electrode. And we call this all optical electrophysiology. And compared to the previous versions using rhodopsins, for example, the quasars, the advantage of our version of all optical electrophysiology is that the dye emission is significantly improved. It's much brighter and requires less laser illumination. We could also extend this all optical electrophysiology measurement to record synaptic transmissions. For example, we have two neurons under the microscope. The green one expresses a light-sensitive ion channel, and the other one expresses our voltage indicator. Traditionally, to measure the synaptic transmission, it requires putting a pair of electrodes, but we want to get rid of this very tedious and experimental setup. So we simply do the all-optical electrophysiology by illuminating the sample with a blue light and recording in the neighboring cell. We could indeed see a induced firing of action potential, and we can see the details of this waveform. When we throw in the APV to block an NMDA receptor, you could see a slight change in the action potential waveform, but then it could still, we could see a very robust transmission across the synapse. When we add in a different inhibitor, and VQX, to block the AMPA receptor, it could completely wipe out the synaptic transmission, as has been reported in the literature. And by doing the subtraction, we could see that our reporter has sufficient sensitivity to tell us the relative contribution of two types of receptors to the synaptic transmission. And we are currently improving the sensitivity of the voltage indicator. At the same time, we've been thinking whether we can extend this method from recording in the culture cells to brain slides or ultimately in vivo. So current version has several limitations. One is that it requires two-step labeling to install the fluorophore. And in the first step, we call the probe incorporation mediated by enzyme, or PRIME. It requires delivery not just of small molecules, but also a 37 kilodalton enzyme to do the task of site-specific protein conjugation. And diffusion of enzyme in the tissue can be very challenging whenever the size exceeds 10 kilodalton. So we want to use a very smaller probe, and also by replacing the two-step labeling with just one single step. So the idea is to try to assemble a conjugate in the test tube and simply expressing a engineered rhodopsin in the cell. Then we just label the sample using a previously prepared fluorophore conjugate to complete the labeling step. We chose nanobody because of their small size and high affinity towards its antigen target. We call this method nanobody-based voltage indicator, or NAVI. And compared to HVI, this NAVI features only one-step labeling. It's much easier to operate inside in the cell, in the lab. And we can now only need to deliver a 10 kilodalton nanobody instead of a very bulky enzyme. We want to test whether this method would indeed work, because using the nanobody requires the distance between the fluorophore and the retinal is slightly longer than simply conjugating the fluorophore directly onto the protein. This would lead to a decrease in the voltage sensitivity. But if we could carefully choose the fluorophore conjugation site, we might still achieve very high sensitivity. In the very first version, we will conjugate our fluorophore to one of the terminus of the nanobody. And indeed, this could work. We achieved about 10% sensitivity in terms of delta F over F, the fractional fluorescence change in response to action potentials. Then we gradually changed the fluorophore position of the conjugation site. Ultimately, I identified a sweet spot that could yield 25%, almost more than twofold improvement in voltage sensitivity. And the nanobody conjugate fluorophore could label the culture neurons very nicely. You could see a very clean membrane localized pattern under the fluorescence microscope. This all worked quite well, and we can achieve very high voltage sensitivity. Currently, we are still in progress of optimizing the system, but we have already shown that indeed this method could work in the brain slice. We will inject the mice with a AAV encoding a synapsin promoter-driven NAVI, and after a few weeks expression, we will cut the brain slice, and we can deliver our reagent to achieve specific labeling on the neurons expressing our voltage indicator. And unlike the previous HVI version, the NAVI seems to work quite well with the brain slides, and we hope to further optimize the labeling and voltage sensitivity, so eventually could work in biologist labs. In another direction, we are also curious whether we can leverage the high spatial resolution and the high throughput of measurement of using voltage imaging to try to understand some how the neurons will or why neurons will behave differently when they are stimulated with the chemicals or when they are treated in the gene knockdown experiments. And when doing voltage imaging, we could see a very heterogeneous behavior in the neuronal population. The same drug could elicit a very different response in the neurons. Of course, some of these might be due to the circuit level behavior, but also some of these could be attributed to the expression profile of each individual neurons. And the idea is to apply voltage imaging, which is which has high spatial resolution and very high throughput, to allow us to observe a cell-specific response or the phenotype. Then we would like to take individual neurons and run single-cell sequencing analysis to identify the genotype, and ultimately linking the phenotype with the genotype to understand what's the underlying mechanism to cause this differential response to drug treatment, gene knockdown, or metabolic shock, oxidative stress, etc. And one of my graduate students designed a microscope especially for this task. We will start with a wide field imaging to identify the regions of interest. Then we will zoom in onto these regions to apply voltage imaging using our very sensitive HVI voltage indicator. After that, we will aspirate the target neurons to run single-cell sequencing. It's a very straightforward pipeline. So here is some preliminary data. Under the microscope, we could observe the expression of our voltage indicators across the field of a few millimeters by a few millimeters. We could zoom in into these regions to identify each individual neurons. This to highlight the high spatial resolution of the setup we are using. And we did a pilot experiment where we expressed a light-sensitive ion channel in a subpopulation of the neuron culture. And under the microscope, we could image their morphology, and we can zoom in different regions to shine blue light to see how they will respond to the light stimulation. Indeed, some of the neurons just doesn't care about the blue light illumination, and some will respond by firing out extra potentials. We will apply this single cell picking using a pipette-based method that we have optimized in our lab. And then we will run single cell sequencing analysis. And we could see that compared to the cells not responding to blue light, which we call the control here, and for all the cells that respond to the blue light, will have a higher expression level of the sheriff protein, which is the light-sensitive ion channel that we introduced. So indeed, this method can sensitively tell the difference between their behavior or the phenotype, and can allow us to resolve the genotype at a sequence using single-cell sequencing. To briefly summarize my presentation, we have created a palette of bright and sensitive indicators that uses the enzyme-mediated ligation and bioorthogonal chemical ligation methods. And we are now in the progress of creating nanobody-assisted labeling so that we could eventually get this to work in the brain slice. And we have achieved multiplexed imaging, and we can also pair our voltage indicator with optogenetic activations. And once we have done that, we can extend our method to try to apply a video-guided cell selection workflow to run what we call HVI sequencing, HVI-seq, to understand how the neurons would behave differently in response to the same stimulus. And with that, I would like to thank you for your kind attention, and I'm happy to take any questions. But I also want to thank my lab members for their very hard work, and our collaborators, particularly my colleague Peng Chen from Peking University, who has been an expert on the bioorthogonal chemical ligation. And we have been collaborating on developing chemically assisted construction of HVI. Thank you very much.
Great, thanks, Peng, for a good talk. Now, John, question time.
Hi, John Knott here. Sorry, my camera's kind of wonky. Really cool stuff. We got in the last bit about combining the imaging with sequencing. It looks really cool, but I would imagine your throughput's going to be limiting. Have you thought of ways to increase the throughput so you're not just picking cell by cell?
Yeah, so thanks for the question. So right now we are looking for a very dramatic change in the phenotype. For example, when as the example I've shown, so that we don't really need to pick up so many cells to understand the underlying mechanism. I think the throughput is potentially one limitation of the method. So it could depend on what kind of question we are trying to ask. So right now the throughput is limited to about a dozen or so cells on a single day. So this is not meant to be a very high throughput method for screening, but to try to pick up the cells with a very unique pattern of exponential firing or very unique electrophysiological properties.
Right. No, that's totally fair. I'm just wondering, looking forward, if you wanted to expand it, whether there's a way to lay down some fiducials and then to take an en-face section of the slice and then to do, say, spatial transcriptomics. Might be asking a lot, but yeah.
Yeah, what we have proposed here is not really the best way going forward with spatial transcriptomics, because we are only reading out the information from a handful of cells. And if you would like to profile, for example, every single cell in the slice or under the field of view, then there has to be other ways for doing that. We are only picking up what we call the winners in the neuronal population to do the analysis on that.
Yeah, great, cool. All right, thanks very much. Really, really nice work.
Thank you.
Okay.
Peng, yeah, please. Yeah. Please. Yeah, go ahead.
Hi, this is JY from Georgetown University. So it's great work and a very great signal to noise ratio. So my question is, in some of your figures, you show the upward means when you have action potential, the light become brighter, and in others, the light become darker. Are these two different mechanisms or two different threads?
Oh, you mean here?
Yeah, the polarity of the signal of the light. Some you increase the light intensity when you have a depolarized. Some are decreased when you depolarize.
Yeah, thank you for catching this. Let me pull this slide. So the way the sensor works is that when there's a depolarization, the rhodopsin will absorb more, so causing more quenching of the dye. So upon depolarization, the dye will go dimmer. So that's the same throughout all my presentation. But there was a mistake I will explain later. So for example, in this slide, you can see the y-axis we labeled as the negative 30%.
You have to share the screen. I don't see the screen. Yeah.
It was showed. Yeah, it's showing the screen. Can you see? We can see the screen.
I don't see it. Okay.
Yeah, we can see.
Yeah. So we label this as a negative 30%. So the fluorescent signal actually will go down. So we simply just flip the signal for this presentation. But then there is a mistake I just saw as you mentioned it. So for this one, the navy, we labeled as negative 10%, but this is really just a 10%. So this is a mistake. So the fluorescent trace you see is that the typical recording that fluorescence will go down whenever there's actual potential spike.
So there are some discussions on how this light change. So if you are working with the tissue, let's say you stain many neurons at the same time, then the background is very high, and then if you if you dimmer the fluorescent when there's action potential, it's actually much smaller fractional change than you brighter the
exactly
current for. Yeah. So I was wondering if you are working on the the dye with the other directions.
Yes, thank you. This is indeed a serious problem that's been recognized by the field. So shortly after the Voltron has been published, the guys at the Geneva farm has worked on the positron, and you can switch the polarity of the response so that upon depolarization, the signal will actually go up. And in my lab, we've been also working on this. This requires mutating the rhodopsin and mutating several key residues involved in binding to the proton or proton transfer pathway and do the screening. It's very hard to do this in a more intelligent way. For example, structure-guided mutagenesis or even computational modeling will not really tell us useful information which are the residues that we could mutate to invert the polarity of the response. So we have to do a lot of screening. And so currently we do have a few versions with inverted response, which means they become brighter. And the idea is exactly as you said, we want to have very low background level when the neurons are quiet, and we can only be bright when they fire action potential. This could increase the signal-to-noise ratio, particularly for tissue imaging. For cultured neurons, this may not be a big deal because there's very little overlap between the neurons. But in brain slide, the issue becomes much more significant. Currently, the positive going versions are less sensitive compared to the versions I'm presenting here, which is also true for positron. Positron is less sensitive than Voltron. And we're still working on that. I think eventually this will be the direction to go. And hopefully in the future, we could have a more sensitive positive going version. Yeah, thank you very much.
Well, thank you very much. Very impressive work.
Okay, thanks, Jian. Any more questions? Okay, I might have a last question. So quick comment, I guess Jane also already asked this question, most neuroscience wondered. So in order for this in vivo labeling, what kind of, you still need to put into ligand, right? It's like CA5, CA3 ligand on the slides. So is that possible to make it a pure genetic encoded multi-sensor some way? Hybrid, or yeah, not so, hybrid, right?
Right. So actually, in my lab, we have several parallel projects. One including completely genetically encoded voltage indicators, GAVIs. Sure. In terms of sensitivity, HVI and also the Navy is more sensitive, has much broader, higher dynamic range compared to the GAVI. So I think it depends on the applications. For example, for the HVI-SIC, when we are dealing with cultured neurons or brain slice, maybe HVI and the Navy is the way to go because of their improved sensitivity. But for live animal experiments, I completely agree. By simply injecting the virus and to have a genetic coded sensor expressed without having to do any chemical ligation or sample delivery or reagent delivery is the most ideal scenario for most of biology neuroscience labs. So we're still working on the GAVIs, and we took a different strategy from the previous approach by trying to insert the fluorescent protein in different locations, including the loop regions of the rhodopsin. And so far, we have been successful. We have successfully identified one region that can give very high sensitivity. This requires a lot of trial and error to do that. So maybe in the future, we could present a improved GAVI for that. Thank you.
Great. That will be a huge leaps to the neuroscience field. Great. Looking forward to the new progress of your lab. Great. Okay, if there's more questions, like thanks Wei and Peng for great talks today. Okay.
Yeah.
Thank you.
Thank you all for great talks, and see you all next week. Right. Thanks, Wei.
Thanks, Wei.
Great.